So we were discussing about the lipid bilayer. The bilayer is basically is due to the lipid nature because it contains two parts. First one is the upper one is the polar one and the two unsaturated fatty acid chain. As we can see here, the circle is the head part or polar part, or we can say hydrophilic part. They love to arrange themselves toward the water, toward water. Okay, and these two are the tail part, that is hydrophobic part. They tend to remain away from water. So why the lipid membrane in the plasma membrane structure is bilayer, why not monomer? We have already discussed that the plasma membrane separates the cytoplasm with the outer environment, and cytoplasm is the aqueous environment, and the outside of the cell is also the uh, aqueous environment. So if the monomer, this is the bilayer, as we can see the upper part, or we can say the outer leaflet. Okay, this one is the second layer or inner leaflet. These are toward the cytosol and these are toward the exoplasmic space. Okay, so these are the two lipid layer which are arranged in such a manner that they are attractive, means they in the cytosol region there is a water. Okay, let me take some another pen. Yeah, there is a water. This is the cytosol region. Okay, this is the outermost region, there is also water. So see, the hydrophilic part is attached toward the watery side. The hydrophilic part is attached to the water side, either in the exoplasmic space or in the cytosol, or we can say the cytosolic phase. So in both the phases, the lipid structure or the polar part of the lipid structure is arranged toward the water. And these, let me take some another pen. And these tail, okay, are you able to see or not? Okay, these tail, which is marked with the green, are toward each others, means they have attraction toward themselves, not toward the watery part. Now we were talking about if the lipid layer is the mono layer in the membrane, then what will happen? Suppose this is the monolayer. Okay, this one is the monolayer. So outside the layer there is a water, and also the inside of the cell there is a water. Now these are the polar part. These are the polar part of the lipid structure. They are attracted toward the water. Then this bond is okay. This bonding is okay with the outer membrane, or we can say the outer side of the plasma membrane. But these tails will never be toward the water. They always tends to repel themselves. So this formation of monolayer is not possible because inside the structure there is an aqueous environment of the cytoplasm, and there needs to be some polar head or polar molecule is should be there so that the tail part should be stabilized. So in this way, so the monolayer is not attached. Therefore, the lipid layer is bilayer, like this way. The first layer of the lipid with its tail, and second layer of lipid with its tail. So this one is the cytosolic part, or we can say site, and this one is exoplasmic site, that is outside the cell. So these structure, that is the polar one will attract with water easily, so no problem with the attachment. The polar head of the inner leaflet is also attached, means attracted toward the water, no problem. And the tail region is also attracted with the each other. We have already learned the phenomena like, dissolve, like. So the polar part will be toward the polar region, and non-polar will be toward the non-polar region. Now in this way the lipid is arranged. So we have talked about the lipid bilayer, the first and the second layer, but they are the water this side, water this side. Okay, let me erase something. Yeah. Now there is a water outside, water inside. This stabilizes each other by reacting, or we can say by heading toward the water region. Now, what happened to this region? These are still facing toward the water. Suppose, then what happened? Okay, we'll discuss in the next slide. Okay, suppose there is a beaker filled with water. Okay, and the lipid layer will tend to arrange like this. Polar molecule will be toward the water region. Now, if we add more water, then what happens? They tend to arrange like this. Now, if we add more water, then they get surround themselves like this and form a lipid layer like this. This is structure basically, sorry, the structure will be arranged like this. This structure is basically known as liposome. That's why lipids are arranged in the bilayer form, because they tend to arrange themselves and want to be away from the water part. That is, the polar wants to interact with the polar region, and the hydrophobic region wants to interact with each other. In this way, okay. Now let's switch to next slide. Yeah, this is the case. This is the lipid bilayer sheet, as we have already seen, and this is the liposome structure. This is the half structure of it. If we see the polar head outside and inside, it's like this only, this structure and this structure. Okay, the polar is like this. So when the water concentration is more, the lipids are arranged in the liposome structure in this manner. And there is a single layer lipid sphere also available, like in the case of the missile. So in this way, the lipid get arranged to the bilayer form. Also, um, yeah. Also, we get to know that the major, the model structure, we can say the model is structure for the discovery, or we can say for the analysis of, for the detailed experiment of plasma membrane. The model structure taken for detailed experiment of plasma membrane was mammalian RBC cell. Why mammalian RBC cells were taken? First of all, it do not contain nuclei. Second, not containing any first of all, they do not contain nuclei, and second, they do not contain any internal organelle, we can say, or any other membrane. Therefore, the mammalian RBC can represent a source from which pure plasma membrane can be easily isolated for biochemical analysis. So this was proved by the experiment also, that the lipid bilayer was in the bilayer arrangement, not in the monolayer arrangement. This was all about the arrangement of lipid, the structure of uh, fluid mosaic model, its composition, and the lipid bilayer, how the lipid get organized or they are arranged in a plasma membrane structure.